So, welcome to this fourth lecture on overview of immunology and these individual cell types that are participating in the immune system. And we looked at natural killer cells before, and natural killer cells recognize specific signals on the surface of the target cells. There are two types: activating ligands, which tell natural killer cells via a natural killer cell receptor that there's something wrong, something maladjusted inside this cell, such as a viral infection, or that this cell has become cancerous. In addition, the normal MHC1 expression on the cell surface is altered in malignant cells or in infected cells. And natural killer cells recognize the absence of MHC class 1. It makes sense that malignant cells, cancer cells, and virus-infected cells downregulate the expression of MHC1 on the cell surface, because the role of MHC1 is to report the presence of foreign antigens on the surface in order for cytotoxic T cells to come and see this through their T cell receptor and then destroy the target cell. So it makes sense for a cell expressing foreign antigens to be able to downregulate MHC1 so that the target cell can't present those foreign antigens. You might ask what's foreign about a cancer cell? The cancer cell is part of our body. Yes, it is part of our body, but the gene expression pattern of this cell will change drastically to express antigens that it didn't normally express. So MHC1 expresses self and foreign antigens. Self antigens do not normally activate our immune system, because in the maturation process of T cells and B cells, they get tested against all the host antigens. So and any T or B cells that have receptors that could recognize our native antigens are deleted. So the only things that get recognized as something foreign. Malignant, cancerous cells, they express unusual epitopes, we call them neoepitopes, and which are markers for cancer. So natural killer cells that have an inhibitory receptor, such that if everything is fine, if the amount of MHC1 is normal, then the natural killer cell is inhibited. But if this inhibitory receptor doesn't find any MHC1, then this is one of the two steps which can result in activation. But the other one is the specific recognition of the activating ligands, which are produced by infected or malignant target cells. So now we're going to start looking at the natural killer cell and how it goes about killing the target cell. And first of all, if we look down here, we have a time-lapse photography of a target cell here and a natural killer cell. And as time progresses, so the natural killer cell starts to interact with the target cell. It's recognized that cell. Now we have 160 minutes, 240 minutes. And what we're seeing here at 240 minutes is the successful destruction of the target cell by apoptosis. And in the diagram here, we're seeing an overview of the beginning of this process, and the result of the action of the natural killer cell matches this here. So this is the apoptosed cell. Here we have the natural killer cell, and now what's shown here is two different mechanisms. There are two mechanisms by which the natural killer cell produces this apoptotic effect. Everything comes in twos, it seems, doesn't it? First of all, there are two mechanisms for the natural killer cell to identify its target. But now there's two different things.
صراحة يا دكتور الصورة والصوت راحة.
الصورة راحت؟ أيوة. عندي الصورة موجودة. That's very strange. Could it be a
الصورة راحة.
Sorry? You see, in my
الصوت والصورة راحوا مرتين ثلاثة.
اوكي، هذا بيكون الخط يعني عندك يا زلمة. رجاء انت وين، في رفح؟ Okay, so students are having a problem with the sound coming and going. It's going to be on their connection side, which is why they will benefit from being able to review the lecture later once it's recorded onto YouTube. Okay, so we have two different receptors which are able to deliver deadly signals to the target cell. One of them is something called the death receptor dependent killing. And the other is granule dependent killing. So two different methods of killing. On the next slide, we will see how these two different methods work. And as I said, it's just a coincidence that everything seems to be able to work in twos. Inside the target cell, there are also two intracellular pathways which result in this apoptosis. So there are two methods of delivering the killing signal, and each method stimulates both of the intracellular pathways which lead to apoptosis. So here we have the natural killer cell. Here we have what's called the Fas ligand and the Fas receptor. And then we have the Fas associated death domain. So this is the death receptor. As the name suggests, it's a domain which, after stimulation with the ligand, the activating signal. It's a strange thing that on the natural killer cell it's called the ligand, and on the target cell it's called the receptor. It's just a matter of perspective, whether you see the thing from the perspective of the target cell or the natural killer cell. So expression of this depends upon the state of this target cell, and this is a signal which will tell the natural killer cell, which will enable the natural killer cell to activate that receptor. So we've got the presence of the receptor, but the receptor must be activated by a natural killer cell in order to lead to this apoptotic process. And this apoptotic process has two different pathways, as I mentioned. This one, which is called the BID pathway, which goes via the mitochondria, and then via release of cytochrome C, and the formation of a body called the apoptosome. It's a complex of proteins which function as a unit in order to result in apoptosis via effector caspase activation. So this is one pathway going via the mitochondria. Another pathway goes directly from the death receptor to the effector caspase activation. There's a lot on this slide, but it will all come together gradually. So primarily now, in the next slide, we'll look at this intracellular pathway in more detail in the next slides. But now the objective of this slide is, to introduce these two pathways, but to focus on the methods by which the natural killer cell is able to stimulate these pathways. So the death receptor is able to stimulate the mitochondrion dependent pathway and the mitochondrion independent pathway. So mitochondria, you all studied mitochondria, and you probably know that this is where the Krebs cycle takes place, and this is energy production. But the mitochondria has other vital functions in controlling cells. Now, the other weapon that natural killers have is the cytotoxic granule, and they have a lot of cytotoxic granules, which, after recognition of the target cell, these cytotoxic granules exocytose and release their contents. And they contain two types of things. One of them is they contain things called granzymes. And these granzymes are bodies which are a conglomeration, an association of multiple different types of protease. And these proteases get into the cell. And they get into the target cell by way of a special opening which is made into the target cell membrane by a molecule called perforin. As the name suggests, perforin, from perforate, to make a small hole. And perforin is also present in these granules. So the granules release perforin, which opens up a small hole in the membrane, allowing these granzymes then to enter into the cytosol of the target cell. And the granzymes also have the ability to activate the two pathways of apoptosis. Apoptosis is programmed cell death. And the two pathways again are the BID pathway, which works via the mitochondrion, and the mitochondrial independent pathway. So we've got these two weapons present on the surface of the natural killer cell, one which stimulates the death receptor, and the other one which introduces granzymes, which will function either in the mitochondrial dependent pathway or independent pathway. Finally, at the end of these two pathways is activation of effector caspases. And if you're looking closely at the slide, you'll see the word caspase appearing more than once. You see it up here, caspase 8. And if there's a caspase 8, then you may imagine that there were other caspases with lower numbers than 8. There is caspase 9 in the apoptosome. And here we have caspase activation. So there is actually a whole collection of proteases which work together in a systematic way in order to result in programmed cell death. And it makes sense that a protease is involved in programmed cell death. What's required here is to destroy the cell without stimulating widespread inflammation in the body. So apoptosis is a non-inflammatory form of cell death. That's what apoptosis is. And the activation of these caspases basically results in now the presence of proteolytic caspases, which will go and digest proteins within the cells. And the end result of all of this will be that the cell contents and anything which could be an inflammatory signal will be trapped within granules. The cell will shrink, the nuclear DNA will be digested, and the apoptosed cell is a nice picture of an apoptosed cell. It's all shrunk down. This will be digested by a macrophage. So we have what we call this a caspase cascade. Some of you are studying medicine, so you'll come across the idea of proteolytic caspases in various places. In this course, you'll come across the idea also when we look at the complement system, where a series of proteolytic events, one by one, result finally in the effector function. Also with platelet aggregation, with thrombosis, you will see how a proteolytic is, not in this course, but in other courses, how proteolytic cascades are also involved in blood clotting. Okay. By the way, also cytotoxic T cells also release some perforin. Cytotoxic T cells have the word CD8 next to them because they express a co-receptor called CD8, which recognizes the MHC class 1. But that's not our subject today. But the link here is perforin. I've already talked to you about the death receptor pathway and its activation by Faz ligand on the natural killer cell. At the molecular level, trimerization of the receptor occurs. This is a theme. Once you appreciate this once, this will apply for dozens of different cell signaling pathways that you might see either in immunology or in other aspects of cellular physiology, where ligation or binding of the ligand results in forming a multimer of receptors, usually a dimer. So this trimerism is an unusual case. In the case of the Faz receptor, then 1, 2, 3, it forms a trimer. It then recruits these death domains and then recruits caspase 8. And that also is a common theme. So a common theme is that presence of ligand causes multimerization, so dimerization and trimerization, followed by the recruitment of other molecules to the cytoplasmic part of this receptor, and the recruitment of a whole series of molecules, and even this is a simplification of that story, results in the effector function of that receptor. And this terminology, which I mentioned at the beginning of effector, that's something which you see over again: effector function, effector cell, effector molecule. Because cell biology, which is at the heart of immunology, is so complicated that we need to break it down into small black boxes. So even without appreciating or knowing, and we're still discovering many, many things on a daily basis about the molecular basis for immunophysiology or any other aspects of our how our body works. Okay, so that's the idea of effector. Okay. Now, here, I'm going to start looking at a little bit more detail about the cellular pathways of apoptosis. So we finished now, almost finished with the role of the natural killer cells weapons, but we will look a little bit about how the granzyme actually interacts with BID, and then from BID we'll continue looking at this pathway. We look at this pathway as well, but we'll focus a bit more on this pathway. BID stands for BH3 interacting domain death agonist, and that's common that we have unusual names, and sometimes these names are indicative or meaning something, but commonly they are an acronym derived from some description of what we know about the function of this protein. Talking about themes, BH3 interacting domain, we find that there's considerable homology within the cell, whether an immune cell or any cell. There's considerable homology between the many different proteins that are functioning there. And proteins are often built up from domains like blocks of Lego. And one of these domains is something called the BH3 interacting domain. Here we have the word domain, death domain. So there are thousands and thousands of different proteins in the cell, but many of them are built up from a limited number of domains. There may be sequence differences between the different proteins in terms of their domain, 90%, 95% sequence similarity at the amino acid level, but enough similarity to consider them to be cousins belonging to the same family with similar structure and similar functional properties. So you just need to know that BID is an intermediary in the apoptotic pathway that proceeds via the mitochondria. BID is part of a family of proteins. So as we have families of domains, we have families of proteins. Something called the BCL2 family contains many, many members, most of which are involved in activating apoptosis via the mitochondria. And one of the key intermediaries, one of the key parts of this pathway is this molecule here called BID. When we say it regulates the permeabilization of the outer mitochondrial membrane, it actually gets cleaved by either the granzyme or caspase 8. And once it's cleaved, it can bind to the outer mitochondrial membrane. And that's how it regulates the permeabilization. Permeabilization means to let something be permeable, to let molecules to pass in and out. Lipid membranes are impermeable to all except certain very small uncharged molecules. But once BID binds to the mitochondrial surface, many things happen, but the result finally will be that something can either go in or go out. And that something is cytochrome C, and that cytochrome C goes out of the mitochondrial membrane. So cytochrome C is a part of the electron transport chain, which is involved in, and that's something you're familiar with. When most people think about the mitochondria, they think about the Krebs cycle, the electron transport chain. Cytochrome C is part of the electron transport chain needed for energy production. But if the cell is going to kill itself, if it's going to undergo apoptosis, then it doesn't have much need now for energy except just to finish off the suicide process. Cytochrome C is released. And it turns out that when people first identified that cytochrome C is being released from apoptotic cells, they thought perhaps this is a part of just a side effect of the dying cell. But actually, cytochrome C released from the mitochondria is a major component of mitochondrial-mediated apoptosis. That's how the mitochondrion is involved in apoptosis. It releases cytochrome C, which combines with several proteins, actually, amongst them caspase 9, to form a complex star-shaped body called the apoptosome. Some means body. So we see this word quite a lot in naming things in the cell. This is a body within the cell which does apoptosis, causes apoptosis. And this apoptosome signals effector caspase activation. Okay, so the next slide is going to look at the cleavage of BID. And then the slide after that is going to look at what happens to BID after it translocates to the mitochondria, and how viruses may interact with it. Let me just pause for a moment because I've received a signal which is making me a bit suspicious. Okay. Hello? Checking my settings. Okay, seems fine. Everything seems fine. All right. Okay. I'm going to okay, I'm going to continue. I wasn't sure to continue or not because I've lost my Gazan students, but hoping that the lecture is being recorded, they will be able to follow the lecture again on YouTube later. So apologies for disruption. Okay. Backs and back are molecules which are recruited to the mitochondrial membrane after it binds to the mitochondrial membrane. Actually, I will stop the lecture here and I will open up a new lecture in a minute, and that will give me a chance to check that the students didn't get lost because of a problem from my side. I assumed it's a problem from their side. Okay, so stop recording.